Muted. Okay.
Yeah.
Anyhow, imagine a future where kids have this innate immune defense against cancer. What's it called? We already know about its immune system. Um, right. And the future of therapies are going to be reshaping the delicate dance in immunoediting in the future. And it'll be imperative that we identify biomarkers that can direct the course of treatment, perhaps bridging the gap between reality and the twilight zone. Um, Most of us are already familiar with the anti-cancer immune cycle, beginning with tumor cells releasing antigens. These antigens are picked up and presented by APCs and dendritic cells to the adaptive immune response, and these migrate towards the tissues. They recognize the cancer cells. Hopefully, they kill the cancer. It's being increasingly appreciated, however, that separate to this, there's simultaneously a more localized immune cycle happening at the same time where the cells that migrate into the tissue, so we're talking about dense lymphoid aggregates and tertiary lymphoid structures, have an impact on the effector state and function of the cells that are entering into the tumor. And using complex spatial mapping algorithms, we're going to be looking at the spatial phenotypes of the cells in these areas and how they interact. Of course, to begin, we should note that yeah, cool. A stunning failure of the immune response is that it's incredibly gullible. Most of us, like I mentioned before, are familiar with PD-1 and PD-L1 axis. So tumor can upregulate PD-L1 to inhibit cytotoxic T cells from inducing death in the tumor cell. However, if we administer an anti-PD-1 monoclonal antibody, we can reinvigorate this cytotoxic response and still achieve tumor cell death. And here on the right-hand side, you see a live action shot of this happening in multiplex immunofluorescence. A recent phase 3 trial comparing patients who received only chemo versus chemo in combination with anti-PD-1 therapy showed a roughly 50% increase in median event-free survival, amounting to about 10 months. So we know that this, to some degree, works. In the neoadjuvant setting, in which the treatment is administered prior to surgery, the immune system has an opportunity to become sensitized to the tumor before it gets cut out. And another benefit would be that at the time of resection, we can evaluate patient response to therapy by looking at how much tumor remains on the tissue slide. Speeding ahead. So the amount of response is evaluated on a continuum from 100% residual viable tumor left in the resection to none. And some key terms: if you have no remaining tumor in your resection, you were termed a pathologic complete responder. And here you can see this is a multiplex immunofluorescence image of the regression bed, the bounds annotated in blue. And within that, you can see features of regression, notably these abundant TLS structures containing B cells and other lymphoid cells. And it's been established that there are multiple histologic features of regression that we can see in the tissue, maybe not from this scale, but think fibrosis, formation of new vasculature, foamy macrophages, et cetera. And inherited from the chemotherapy era is this threshold of 10%, which qualifies major pathological response. So here we have, again, the regression bed in blue. And what's left is this chunk of tumor, about 10%, we'll say. But you can still see some features of regression, though not to the same extent, existing in the regression bed around the tumor, indicating that there's been some degree of response. And when you don't have these features at all, and you don't have any regression whatsoever, you're a non-responder, you can see this massive tumor has not been affected by the therapy. If you're wondering what these little green pockets of stain are, it's PD-L1 expressed by the tumor itself. So it's a non-infiltrated tumor. Great. So these are multiplex immunofluorescence images, and why are we so excited about multiplex immunofluorescence? Here's from a study on over 8,000 patients and a whole bunch of tumor types. If you're not familiar with a receiver operating characteristic curve, they're deceptively simple. So on the y-axis is your true positive rate in estimating some metric. In this case, it's response. On the x-axis is the false positive rate. And if you imagine a perfect test you'd be 100% perfect and never false. So you have this little line that would go all the way up to up here, and the area under that curve would be 1 in theory. That never happens. By comparison to, God bless, to PD-L1 IHC, the histological gold standard, tumor mutational burden and gene expression profiling, even multiplex immunofluorescence, this green line showed a more robust, closer to this ideal test association with response. So we know it has a lot of power in detecting these biomarkers. And you might remember Emily. She did a master's degree here some years ago, like one, and she found these really rare cells, CD8 FOXP3 cells that make up not even 3.5% of all CD8 T cells. And on the right-hand side, what you can see is this heat map of AUC values. That is, so the area under this curve, which we plotted. And the greatest predictor of response in these pre-treatment samples was CD8 FOXP3. Interesting. And to get a better picture of what's actually going on, a colleague from Johns Hopkins, Jeff Pesci, we call him affectionately, looked at the interactions of these cells and tried to identify a neighborhood surrounding the CD8 FOXP3 cells. And he developed a model to identify these relationships, whether or not the central CD8 FOXP3 cell was present. And by the end of this, you'll know exactly what a receiver operating characteristic curve is. Here are two more. What's important to note is between the two, they're fairly concordant, which just goes to show that the cells alone are important, but it's also important to be identifying these spatial relationships when trying to evaluate biomarkers. This was all done in pre-treatment specimens from the first clinical trial of neoadjuvant PD-1 in non-small cell lung cancer, and we have the on-treatment resections. And so it's going to be my goal to be evaluating the kind of patterns like these that exist in the on-treatment specimens. Here's my hypothesis. CD8 FOXP3 cells will co-localize with features of a productive immune response in the on-treatment resection specimens of the same cohort that Emily was working with. So the first thing I'm going to be looking at is the relationship between these cells and TLSs, as well to look at the activation states of these cells and the cells in their niche, and then to look at other features of response, like I was talking about the histological features of regression. So what do we have? From that cohort that Emily was working with, we have 35 on-treatment resections from 35 patients. So these are all patients who received neoadjuvant anti-PD-1. From each, we take 3 serially cut sections and we stain them with, we have 3 individual multiplex immunofluorescence panels that are targeting distinct aspects of immune checkpoint blockade. So for example, we have a panel dedicated to the PD-1, PD-L1 axis markers of regression. So we'll be looking at B cells and vasculature, and we have a third panel dedicated to activation states, transcriptional activators of T cells. And using AstroPath, our semi-quantitative, or sorry, semi-automated platform for digesting multiplex images, we mapped over 50.5 million cells from these 105 slides. These get loaded up into a database that we interface with through a Python API. My first aim, little refresher, was that we're talking about tertiary lymphoid structures. And first, we need to be able to identify them. And then beyond that, we can look at correlations that exist with and around them. And to do that, we have, like I mentioned before, 2 panels that might be helpful. If we want to ask the question, do CD8 FOXP3 cells correlate spatially with these structures? Here's an example of a tertiary lymphoid structure that you can see from our multiplex panel. These 3 markers are specific to this panel. These 4 are common. And when we combine it with the second panel that looks at markers of regression, including the vasculature and the CD79A, we can superimpose them on a spatial coordinate system and look at where these things localize. So that's down the pipe. But a very simple analysis would just be, broadly speaking, do these cells correlate with these features? So here's new data, fun and exciting. CD8 FOXP3 cell density on the y-axis. And maybe this is a hard sell because that's like the loosest correlation I've ever seen, but it's pretty significant according to the p-value. And then on the x-axis, we just have purely the number of tertiary lymphoid structures you see in the tissue, and there seems to be some kind of relationship. So more to it that we will investigate. Obviously, just inflammation itself would be associated with these things, but I mentioned as well that we want to look at the different types of cells. So when we're looking at things like CD8 is a marker, CD3 is a marker, we lose a lot of the heterogeneity in the phenotypes of these cells. But by including a multiplex panel that looks at transcriptional regulators of, say, exhaustion and memory, we have the ability to look take a more detailed look at these cells. And if you're not already acquainted, allow me to briefly introduce you to EOMs and TBET. They're transcriptional regulators of various T cell genes, including PD-1. This is from one of many studies that found an association between the relative expression of EOMs and TBET in the activation state of T cells. For example, here they show that a high EOMs to TBET ratio is associated with an exhausted phenotype, whereas a lower ratio might be associated with a memory phenotype. And we can try to observe these in the tissue. So here is a truncated version of one of our panels. I've only highlighted the stains that might be helpful. And if we look closely, maybe we can identify this full throttle TBET cell as a cytotoxic T cell, and these with middling EOMs to TBET ratios as perhaps memory T cells. Of course, conjecture and just very eyeball measurements, but we can do this statistically by looking at averages across the whole tissue. What's really interesting, though, is the stark difference between this panel and this panel. Obviously, there's a lot more EOMs expression. And so we might be able to infer that there's a lot more exhaustion occurring. And this is something that will come up later, where certain pockets in the tissue have different niches that behave differently. We will return to that concept. But first, the cells have to get into the tissue for us to see them. And from a broad perspective, can we make any generalizations about patients who respond or don't respond based on the amount of infiltration that they have, with a particular focus on CD8 FOXP3? So here now, this plot is showing on the y-axis the log transformed cell densities, and then we have a number of different cell lineages. This black line is showing the trend between medians in two groups. Those two groups are patients that achieve major pathological response, so they're left with only 10% residual viable tumor. Patients that don't achieve. And there's no statistical significance between the two. And if we look at the other phenotypes, that seems to hold true. CD8 has been cited many, many, many times in the literature as being a predictive at this level of response, or sorry, indicative. But looking at the non-significance overall probably tells us that we need to look deeper, and we need to look at what the cells are actually doing rather than at the whole level of a whole tissue. So we tried to add another dimension to the data. Here now we're looking relative to the tumor boundary, and we look 0.5 millimeter within the tissue, 0.5 millimeter without. We modeled the probability as a function of density across all the tissue samples that we have for discrete distances from the tumor. And here what we can see is that in these 25 pretreatment samples that Emily was working with, the cells seem to abut at the tumor edge, fail to penetrate, and we have very poor infiltration into the tumor. However, if we look at responders, we see a completely different story where the infiltrations massively increase relative to the pretreatment samples. And somewhat similarly, if we look at patients who didn't even achieve 50% MPR, we still do see an increase, but it's nowhere near the increase that we see for patients who achieve a response. And it might also be worth noting that perhaps patients who do not achieve a response, their cells are excluded from entering the tumor because we see this buildup right outside the tumor and fails to penetrate. So even this is still a cursory overview of what's going on in the tumor microenvironment, because we could get much more granular, and we plan to do so. Oddly enough, a lot of this spatial statistics were developed in geography, which makes sense when you think about it, but like, how often do you think about geography? And what we plan to do is model the cell-to-cell interactions to be able to implicate pathways that we might associate with features of regression or features of response. And to explain a little further, the concept of spatial autocorrelation describes the tendency for like things to occur in similar places spatially. So a little quick experiment would be all the students are sitting together and the PIs are in the front. You're more likely to be sitting next to somebody you know than you don't know. Moran's I is a statistic that was, the formula for it was developed in the 1950s, and we can use it to evaluate the degree to which a variable correlates with itself in space. But before, I was mentioning that you can take two different looks at the same tissue and see two different phenotypic patterns occurring, two different niches. It might be of use to be able to evaluate the relationships between variables, i.e. cell types, in different pockets of the tissue rather than across the tissue as a whole. And there are multiple algorithms akin to this that achieve that. And then, dipping our foot back into the twilight zone. I was in Stauffer Library, and there's like a wall of books on geospatial statistics, and about half of them have my name on them. So, like the egotist I am, I ripped them off the shelf, right? And this is crazy. So he's a huge proponent of this method, but also he did his bachelor's like 20 minutes from where my grandfather grew up in Aberdeen, and then like 2 degrees in my hometown. So yeah, I don't know. And like, we don't look alike, but secret fourth aim is going to be steal DNA, run a paternity test, and then claim inheritance. So the takeaways, beside the fact that I'm a loon, but CD8 FOXP3 densities in pre-treatment biopsies associated with response to anti-PD-1. However, the role of these cells is mercurial. We don't really understand what they're doing. Maybe by running these spatial models on the on-treatment specimen where we have a wealth of data, like millions of cells, we'll be able to identify the spatial relationships that explain their function. Yeah, and that's it. So.
All right, questions for Michael? Go ahead, Neil.
I'm going to start with an easy one. So the non-small cell lung cancers, these are adeno-and squamous stripes. So you have both of them here, or is it just adenocarcinoma?
I'm glad you asked. The ones we're working with right now are just adenos, but we do have a cohort of squamous tissue that we can evaluate as well.
Just wondering if they sort of separate.
If they do something differently, yeah, that would be interesting for sure.
Yeah, so first and advice, don't do the DNA test. You never know. So yeah, I'm confused about the phenotype of these cells. So I mean, I know a little bit about CD4 FOXP3 positive, double positive cells, but these are assumed or effector cells. Is that correct?
I can try to show if I'm able to. Oh, there we go. Break into the hidden slides. Here we go. So, right. So sort of what you were getting at. We know what a cytotoxic cell does. We know what we know what a regulatory T cell does, and these are antagonizing effects. It's confusing because you look in the literature, and a lot of people claim that these cells are immunosuppressive. And the primary activity of these cells, the method in which they're immunosuppressive, is by killing T cells through cytotoxic action. So we also have RNA sequencing data single cell RNA-seq that we have from that pretreatment cohort, and they found that these cells do have a cytotoxic phenotype. So something in them is driving that, but the literature is pretty confused. I mean, like there's another paper that showed just having these cells alone was associated with poor prognosis, a low density, that's that low line or red line. And then we find the opposite association. So confusing. There isn't necessarily a consensus. I haven't seen a lot of functional assays that weren't done. I mean, there's lots of mouse work, but that's another layer of variability.
Just I must have done single cell analysis on CD4 FOXP3 positive cells in these. I'm just wondering whether what makes one definitely suppress it, the CD4 forms, and this which seems to be affected.
Right. Excuse me, furball. You raise a very valid point. To my knowledge, CD4 cells aren't cytotoxic, so it's a bit of a curveball that you're suggesting.
Yeah, no. I mean,
Okay.
It's a different subset of T cells for sure.
Yeah.
But I mean, thank you.
Sorry, I just
Can you go
Cut
back
your
to the
question short.
slide where you show the spatial relationship?
yes.
Get which number it is, but we'll recognize it when we see.
I think I know the one you're talking about.
Yeah, we
This
have
one.
two, one with and one, okay, yeah.
Yeah.
So this is interesting, but you don't tell us what the other cell types are that you think you're looking for relationships with, so. I'm gonna guess the spiky ones are what, dendritic cells, or is it just random? You don't know yet what they're gonna be.
Right. So the mock-up is just
Anything.
anything, but so we do have these values. We do know the method used to figure out what is interacting with these cells is we take all the interactions that CD8 FOXP3 cells have, and we had a radius of 10 microns. So cells that are very close to these cells, and the comparator was any other cell, and we looked for statistical differences between these ones and those ones. And so we have a list of, I think it's like several hundred of these relationships. It's not specifically one, excuse me, that
Several hundred different other types of cells besides the CD8 positive.
like configurations of cells. So like you can have a single macrophage next to a CD8 FOXP3 cell, and that's considered an interaction.
Yeah.
And of all the ones we interrogated, there were like we came up with like a hot list of the 100 that might be important.
Okay,
Of
of 100 different types of cells.
not 100 different types of cells, but 100 different configurations of the same
Okay,
cell. So you could have like 2 macrophages in a CD8 cell. You could have 1 CD8 cell.
what if you kept it pretty simple, like that CD8 FOXP3 cell? How often it is adjacent to, I don't know, an antigen-presenting cell.
Yeah.
So keep it simple at first, and then see if you can find correlations with that.
How frequently do they interact with these cells?
Or what is the correlation between a high frequency of that relative to a low frequency of that and outcomes or
Yeah,
response?
that kind of gets to the future directions I was mentioning, where we look at how do these things collinearize in space. So rather than looking at neighbors, we can even look at as you move across the tissue, is there a static relationship between these 2 cell types, which answers effectively the same question.
Okay. So you're doing this in particular areas of the tumor, and it was interesting that you're looking at a boundary effect. I mean, this may be very different if you were to look at the tumor draining lymph node, for example, as opposed to within the tumor, adjacent to the tumor, that sort of thing. And perhaps it's not, you don't have data to be able to look at tumor draining lymph nodes at this point, but if you look in that region, either side of the boundary, do you expect to, or do you have ideas, hypotheses about the likelihood of seeing different sorts of interactions? And the reason I ask this is because I'm always fascinated by this idea that there are CD8 cells piling up at the boundary of a tumor when I always figure, well, is that how they get into the tumor? Don't they get in through blood vessels, which wouldn't they be all through the tumor anyway? So,
I mean, two good points that you brought up in there. One, we do have lymph node specimens that we can evaluate. We also have, yeah, so we do, so we can look at that, but we haven't. Previous literature has shown, I mean, obviously the lymph node is just going to be a mess of all these cells. And when our markers are CD8 and like CD79, like those are very broad markers. So when you see all of a sudden that the lymph nodes, like tons of these things, what can you really tell from it? It's going to be a difficult question. But what's interesting is the you were talking about how cells get into the
yeah, how they get inside into the tumor itself, yeah.
and so T-bet when it's in the, I mean, you know this, when a T cell is activated, T-bet will upregulate CXCR3 on the T cell. But obviously, if you have varying degrees of T-bet, or for some reason T-bet is manipulated in some way, or it's homolog in a sense, there's a lot of crossover function with EOMs. If CCR3 is inhibited in some form, maybe you're not going to get into the tumor, and maybe that's something innate to the tumor or the immune system.
Well, I'm assuming that stuff that piles up at a boundary is encountering some kind of
Some resistance.
extracellular matrix structure that has a barrier effect. But those cells presumably exited a blood vessel or something at some
Somewhere.
point in order to get into that tissue and are moving against that barrier at that point. But there are going to be blood vessels on the other side of that barrier
Yes.
as well, that they could have exited from inside.
Yeah. And so what's the difference?
Yeah.
Yeah.
Yeah. Anyway, fun things to consider. Other questions? Yeah, go ahead.
How big an issue is the fluorophore? So if you start swapping fluorophore, so you're getting the same results.
Right. Yeah. So anybody who's heard astropath has heard, I'm not going to show figures too much, but the whole process of optimizing an antibody takes like optimizing a panel take what, a year? More than that? Yeah, you'll hear about it. But it's a pretty rigorous process, and we don't just use them interchangeably. It goes through all this validation and optimization, and then we keep them. Maybe to your point, another problem that we can have is the quality of the fluorophore itself. So we're looking at a lot of co-expressing molecules. So especially if we're looking exclusively at the nuclei, and we have all these nuclear markers. A challenge is making sure that one's not blocking the other, et cetera. But as part of the optimization process, we set up.
Yeah, no, it's interesting. I mean, it's amazing. Had to find something to pick on.
I did not.
Okay, oh, you have a question? Go ahead.
Thank you, Mide.
Just more curious on just like a general question on like why looking at like certain like sections of a tumor might be more like I guess effective than looking at the whole tumor. Because I know you're looking at 3 slides and then you're seeking like, is there like any sort of like intro like a heterogeneity?
Yeah. So in some ideal world, we can isolate for all the factors that dictate what's going on in the cell, much in the same way that in a city, if you wanted to look at, let's say, I don't know, pizza restaurant waiting times, you wouldn't look at 2-3 restaurants in the same district. You'd look across the whole tissue, but isolate them in pockets. So I don't know. That was a weird analogy, but I guess I'm hungry. Yeah.
But like
Yeah, like 3 million different things that I can't even list. So
Okay, we've run out of time anyway, so thank you very much. So if we can tee up Tashifa's talk next.
it's somewhere there.
Oh, it's great.
Okay, so our next speaker is Tashifa Imtiaz, and so Tashifa also did her bachelor's degree here at Queen's, and she was in the neuroscience stream, but she spent her fourth year project in Dr. Rennick's lab, co-supervised by Catherine Trishkin, I believe, and that's where she became enamored with microRNAs. This was extended into BSc, MSc, one of these combined degrees, which she finished last year, I guess sometime in the summer. But then her fate was sealed because she got accepted into the MD PhD program. So she's with Dr. Rennick and Trishkin for at least another few years. And so congratulations on that. Well, Tashifa seems like a very kind and gentle person, but I've learned that she has a sharp wit, and she can dish it out as well as she can take it in the Rennick lab. So I'm not worried about her at all there. She's doing very well. So take it away, Tashifa. Sorry to embarrass you.
All right, thank you for that somewhat incriminating introduction. All right, hi everyone, my name is Tashifa, and today I'll be presenting my PhD proposal for the role and diagnostic potential of microRNA 375 in gastroenteropancreatic neuroendocrine neoplasms. I really wanted to go first because I did not want to follow up after Michael, but please bear with me. Okay. So neuroendocrine neoplasms, or NENs, are tumors and cancers that arise from neuroendocrine cells throughout the body. They're found within neuroendocrine organs, as well in the diffuse neuroendocrine system, which is located in organs such as the lungs, the pancreas, small intestine, appendix, where you can find neuroendocrine cells. Something special about neuroendocrine cells is that they both have neuronal as well as endocrine differentiation. So that means they can communicate with the nervous system, as well as secrete hormones, and act kind of as sensors for environmental changes. So NEN incidence has increased overall throughout the years to around 6.98 per 100,000 individuals, and this is likely due to increased detection or improved detection methods. And the distribution of neuroendocrine plasms is shown here in the graph, and we can see that majority of neuroendocrine neoplasms are gastroenteropancreatic NETs, making up about 60%. And so GEP-NETs are found within the gastrointestinal region as well as the pancreas. So GEP-NETs arise from neuroendocrine cells in the gastrointestinal tract and pancreas. And here's a list of various different neuroendocrine cells found in the GEP region and different hormones that they release. And based on whether the neuroendocrine tumor is functional, meaning it secretes bioactive hormones, it could be causing any one of these syndromes listed on your right, or it could be non-functional, meaning it doesn't secrete any of these hormones. Okay, so kind of shown in the previous slide, GEP-NENs have a very diverse biology and presentation based on their cell of origin, whether they're functional, meaning they secrete hormones or don't, as well as their differentiation status is neuroendocrine differentiation and their aggressiveness. So some of this is thought to be due to where the tumor, which may have arisen from. So for instance, it could have arisen from a stem cell that differentiated into a primitive neuroendocrine precursor cell. And so if the tumor arose from there, it might be a poorly differentiated neuroendocrine neoplasm. Whereas if that precursor cell continued to become a hormonally programmed neuroendocrine precursor cell, and if the tumor arose from there, we might see a more well-differentiated neuroendocrine neoplasm, known as grade 1 or grade 2 NETs. Another thing to note in this figure is you might see NET and NEC. So the NEC stands for neuroendocrine carcinoma and generally refers to neuroendocrine neoplasms which are poorly differentiated and usually more aggressive. And NET stands for neuroendocrine tumors which are more neuroendocrine differentiated and usually less aggressive. So unfortunately, partly due to the heterogeneous nature of neuroendocrine neoplasms, current diagnostic tools are limited due to their specificity, their sensitivity, their correlation to tumor behavior, and their accessibility. So some of the diagnostic tools used are imaging tools such as MRI, CT, and PET. However, these are limited by their ability to detect tumors in certain conditions, as well as their accessibility for the general patient population. Other diagnostic tools might be proteins and hormones. So some of the ones listed there are chromogranin A, which is involved in the release of those secretory molecules, and synaptophysin. However, these are also limited by their ability to detect neuroendocrine neoplasms, so their specificity and sensitivity, as well as they're able to be modulated if the person is taking medication or they have some other co-occurring conditions, such as they're on dialysis. Another kind of protein and hormone is INSM1, which is a transcription factor often found in neuroendocrine neoplasms. And other ones are serotonin, insulin, different hormones that could, if it's a functional net, for instance, you might see these highly expressed within the blood. Some other diagnostic tools that have been studied recently are mRNA levels or protein levels and circulating tumor cells or DNA. However, unfortunately, especially for circulating tumor cells, though they were initially very promising, they show limited correlation to the tumor behavior or require very precise tools to be detected, which also limits their accessibility. So instead, that's where microRNAs come in. So microRNAs are short non-coding RNAs that negatively regulate gene expression. They do this by binding to target messenger RNAs and leading to their degradation, so preventing their translation into proteins. And microRNAs are involved in a variety of cellular processes, such as controlling apoptosis, growth and differentiation, metabolism in cells, communication through potentially secretion with extracellular vesicles. And unfortunately, microRNAs are also dysregulated in a variety of diseases, including cancer. Furthermore, due to their sensitivity, as well as specificity, stability, and accessibility, microRNAs are considered promising biomarkers. So microRNAs are often secreted into the extracellular fluids, and they're usually associated with either proteins or stored within extracellular vesicles, making them more stable compared to mRNA or other RNAs found in the blood. As well as microRNAs are highly specific to their tissue of origin. So shown here is a figure of different microRNAs that are expressed during mouse embryogenesis, and they're specific to each stage of embryogenesis. As well, they're highly sensitive to different disease stages, and shown here are different microRNAs expressed in NAFLD or non-alcoholic fatty liver disease as it progresses or in ameliorated stage. Okay. So our lab recently showed that microRNA 375 is elevated in neuroendocrine neoplasms relative to site match controls. This figure, so in the yellow or orange, depending on how you see it, are neuroendocrine neoplasms. And you can see that microRNA 375 is generally higher in neuroendocrine neoplastic cases compared to their site match controls. As well as Dr. Zhao Jing, as well as Gina, showed that microRNA 375 targets GAP, which is a transcriptional cofactor and promotes neuroendocrine differentiation in lung neuroendocrine neoplasms. So here, when lung cell lines were depleted, microRNA 375 was knocked out, we saw a decrease in expression of neuroendocrine neoplasm markers such as chromogranin A and synaptophysin, as well as a decrease in tumorigenesis or cell proliferation. However, this was partly rescued by treating with silencing GAP. So this shows that microRNA 375 is targeting GAP to control neuroendocrine differentiation and tumorigenesis in lung neuroendocrine neoplasms. And this is the proposed potential targets of YAP that might be giving the effects that we're seeing. So microRNA 375 might be targeting GAP, which downstream targets epithelial-related genes and tumor suppressors. So instead, now we're seeing by targeting GAP, we see tumorigenesis. As well as YAP might negatively target neuroendocrine transcription factors. So if we have YAP overexpression, we would see reduced neuroendocrine differentiation. So this brings me to my hypothesis and aims. So I hypothesize that miR-375 is a regulator of neuroendocrine differentiation and tumorigenesis, and potentially a valuable diagnostic marker for gastroenterior pancreatic NENs. So I will look at this through three aims. So first, I will investigate microRNA 375 and the YAP axis in gastroenterior pancreatic NENs. Then I will assess if microRNA 375 is a neuroendocrine differentiation marker across GEP tissues with a tissue microarray. And lastly, I will look at if we can establish a microRNA-based liquid blood biopsy for GEP-NENs for diagnosis and monitoring. And kind of a fourth aim as well is to review what the literature says on microRNA 375, whether the literature also says that's a neuroendocrine differentiation marker or oncogene or tumor suppressor. So for my first aim to investigate microRNA 375 and YAP axis in GEP-NENs, Zhao Jing, who is the research associate in our lab, has established two pancreatic neuroendocrine neoplasm cell lines, BON1 and QGP1. BON1 is a carcinoid cell line, so it's a more differentiated neuroendocrine neoplasm, and the QGP1 is a carcinoma cell line, so it's less differentiated, and has done the following experimental conditions. So microRNA 375 knockout, microRNA 375 empty vector control. So that's the typical cell line, YAP overexpression through DOX induction, and then YAP overexpression DOX minus, meaning the control. And then we are going to investigate morphological changes that have happened after these experimental modifications, the changes on cell growth and proliferation, transcriptomic changes that might have occurred, as well as if there is well, we did observe that there's decreased cell growth and increased apoptosis. We want to confirm that through apoptosis assay, as well as look at the expression of various neuroendocrine markers and potentially the impact on cell migration and motility. So these are some preliminary results where we showed that microRNA 375 knockout and YAP overexpression actually decrease neuroendocrine marker expression and decrease cell growth. So shown here are Western blots for expression of neuroendocrine neoplasm markers, INSM1, synaptophysin, chromogranin A. And we can see that when microRNA 375 is knocked out, there's general decrease in the expression of these markers. As well as this shows the colony numbers for the cell lines after microRNA 375 depletion. And generally, after microRNA 375 depletion, cell counts go down. Similarly, when YAP is overexpressed, we see that the expression of different neuroendocrine markers, such as synaptophysin, chromogranin A, INSM1 goes down, as well as cell counts or cell growth go down. And this effect is partly rescued by treating with silencing YAP in the microRNA 375 knockout cells. So when the BON1 and QGP1 cells that had microRNA 375 knocked out are treated with silencing YAP, we see a partial increase in INSM1, chromogranin A, and synaptophysin, all neuroendocrine markers, as well as a bit of a rescue of cell growth. So these are all different metrics of cell growth. So we have the cell numbers, images of the colony counts, as well as a bar graph showing the colony numbers. And generally, when treated with silencing YAP, there's an increase or partial rescue of the cell counts as well. So future directions for this project is checking if we see the same effects in vivo mouse models. And this is thank you to the Greer Lab for helping us set up the experiments for this. And so we are going to be injecting both cell lines into mice models and then measuring tumor size. And once the experiment has reached completion, resecting those tumors, staining for neuroendocrine markers to see the change in potential neuroendocrine marker expression, checking for presence of metastases, especially within the liver, and also doing blood collection to see if we can detect microRNA 375 or neuroendocrine markers within the blood of these mice. Potential future direction is also collaborating with our collaborators at Sunnybrook to look at the effect of microRNA 375 knockout or YAP overexpression in various different models, such as ex-ovo models, and to potentially model the tumor microenvironment and see the effects on angiogenesis. Okay, for my second aim, to assess microRNA 375 as a neuroendocrine differentiation marker across GEP tissues with a tissue microarray. And thank you to Harrison, who is also a research associate in our lab, for helping kind of set this up. So to first start off, I would be collecting formalin-fixed paraffin embedded blocks of GEP-NEN samples from both Queens and our collaborators at Sunnybrook and New York, and then constructing a tissue microarray shown here, where the slides are first annotated and then cored and arranged into a tissue microarray blocks, and then completing in situ hybridization for microRNA 375, and then comparing it with an immunohistochemistry for neuroendocrine markers such as chromogranin A, synaptophysin, and INSM1. So these are some preliminary results from tissue microarray that Harrison is working on, where we can see that microRNA 375 co-localizes with established NEN markers across various neuroendocrine neoplasms. Shown on the left are the names of the various neuroendocrine neoplasms. And then at the top, there's the H&E microRNA 375, INSM1, which is also chromogranin A and synaptophysin, all neuroendocrine markers. And we can see that generally microRNA 375 is positive, where INSM1, chromogranin A, and synaptophysin are positive. And positive is like a darker brown for the proteins and a dark purple for microRNA 375. So for my last aim is to establish a microRNA-based liquid biopsy for gastroenteric pancreatic neuroendocrine neoplasm diagnosis and monitoring. So first, we will be collecting patient samples. This is being done at Sunnybrook as well as here in the Kingston Health Sciences Center, extracting plasma and isolating and extracting the RNA from these samples, completing small RNA sequencing. And so based on that, we will split the samples into a validation and a training cohort because we are going to be proceeding with some machine learning processes. And to assess the generalizability of machine learning tools, we want to have a kind of testing cohort that we can check if our training and our setup was correct. So after we split our samples into discovery and validation cohorts, we will be running machine learning tools to identify microRNAs that are able to discriminate between our neuroendocrine neoplastic and other GEP-NEN disease samples. We will validate the expression of these microRNAs using quantitative PCR, and then train machine learning models to be able to tell, is the sample that it's seeing a GEP-NEN or not? And then finally, we will validate that model on our 20% validation cohort that we put aside. And that validation step will help us see if, first of all, is our model generalizable to new samples that it hasn't been trained on, if our microRNA thresholds are applicable to those samples, and if we need to be using different kinds of machine learning models. So this is preliminary work showing the expression of microRNA 375 in patient plasma for different neuroendocrine neoplasm patients. And we can see that in GEP-NEN patients for microRNA 375 is elevated compared to non-NGP controls, as well as for small cell lung carcinoma, which is also a neuroendocrine lung disease. MicroRNA 375 is elevated compared to non-NEN lung disease. Here you can see for Merkel cell carcinoma, which is a neuroendocrine neoplasm, microRNA 375 does look relatively low. But after reviewing these samples, we saw that these patients had already received treatment, so perhaps 375 levels were affected by the treatment status. Okay, and my last appended aim is to review and interpret the literature on microRNA 375 specificity. So first, I will conduct a search for the literature mentioning microRNA 375. So that's a pretty broad search on any paper that's mentioned microRNA 375, both in normal as well as disease. And then we will review whatever searches come out through abstract and full text review. And then after, we will decide which studies to include as well as extract information. So this literature review is underway. Currently, after the first search, I got about 9,585 results. It is a very broad search. About 5,900 were duplicates. So after removing those, there were about 3,683 remaining to screen. So that's still quite a bit. And then after abstract screening and partway through full text review, about 1,939 were removed because they were irrelevant or didn't really offer insights into microRNA 375 in humans. And so currently we're still at the full text review stage, and there's about 1,390 to screen, and 354 have been selected right now to be further reviewed to extract information. So that's kind of what is going on with the review. And generally, we've seen that there's quite a bit of variability in what people think microRNA 375 might be doing. There's a lot of literature that potentially says that microRNA 375 might be a tumor suppressor. However, there is a decent amount of literature that seems to suggest it could be an oncogene under different contexts. And what we want to interpret this literature as is it a neuroendocrine differentiation marker and specific to neuroendocrine neoplasms. Okay, so just a review on the impact and significance of my three aims. So one of the main goals is to understand the functional role of microRNA 375 in GEP-NENs pathogenesis and in neuroendocrine differentiation. We want to establish microRNA 375 as a tissue and blood biopsy marker for GEP-NEN diagnosis. And then a potential future direction or interpretation would be to identify targeted treatment options for neuroendocrine neoplasms, such as anti-MUIR375 and broad-acting regulatory pathways that are common across neuroendocrine neoplasms and not necessarily specific to just one type of neuroendocrine neoplasm. Lastly, I'd like to thank members of my lab, Dr. Zhao Jing, Harrison, Simona, Gina, as well as my supervisors, Neil and Catherine, members of the Greer lab for helping set up with a lot of the animal experiments, Dr. Gan, Dr. Greer, as well as Danielle for giving me some mice, thank you. as well as our collaborators at Sunnybrook Research Institute, Montfort Einstein at New York, and Rockefeller University in New York. Yeah, open to any questions. Thank you for listening.
Questions? Ready? Go ahead, John.
Yeah, please, I just want to know, you have any ideas, how can we use it? Microarray 375 as a template for therapy.
Yeah, that's a very good question. So I think potentially as kind of how silencing microRNAs are designed, but it's something that's complementary to MIR375. However, like all kind of targeted therapies, it becomes complicated regarding the targeted part. And so what I can imagine is it would definitely need to be packaged inside some sort of lipid vesicle. And that lipid vesicle would probably need to further be able to target the site of neuroendocrine neoplasms, because MIR375 is expressed in other tissues as well as normal neuroendocrine tissues, but at lower levels compared to a neuroendocrine neoplasm. And so much before that can even be a thing, there would probably be preliminary tests to see if we can design a vesicle that is specific to a neuroendocrine neoplasm, if 375 can get in and it doesn't have, if it can knock out 375 to an anti-MIR 375 can knock out MIR375 to an effective enough level, but that was kind of maybe future goal.
I think Monty was the person.
Very clear, the differentiation states of supplements.
Differentiation. Oh, just
Try and repeat the question. I think people on Zoom.
Oh.
might not have that.
so Monty asked to go back to the slide where this one, next one. Oh, this one. Go
You
back to
have
this slide.
transdifferentiation in all states in the CI.
Yeah, that's a really good question. I think there's a bit of contention whether carcinoids can become carcinomas. So carcinoids were the more differentiated one, and carcinomas are the less differentiated one. So if they can de-differentiate into carcinomas, there doesn't seem to be any evidence that that can happen. However, like for instance, prostate cancer can undergo neuroendocrine transdifferentiation, and that forms a very aggressive form of prostate cancer that is resistant to hormonal treatment. Maybe Neil can correct me if I'm wrong. But also in other cases, there have, I was just reading recently about pancreatic neuroendocrine neoplasms. Some are more aggressive than others. And so that paper proposed that very specific insulinoma, which is a functional carcinoid, and undergoes kind of reverse differentiation to become a more metastasis-like phenotype, which is a different form of classification for neuroendocrine neoplasms. So there is a potential, I think, for a more differentiated carcinoid to go into a more aggressive form, but I don't, there isn't that much evidence to show that it goes into a carcinoma form.
Yeah, I was just wondering, when you're looking at the GEP-NENs, why not just look at all NENs? Like I know you talked about the prevalence, like on slides.
Yes, so why not just look GEP-NEN?
Like there's a diverse biology. Right?
Yeah.
Why not just do
All of them. Yeah, I think that's a good. So Tynian asked, why not just look at all NENs? I think the lab broadly will be looking at all NENs. My project will focus specifically on GEP-NENs. And even arguably within GEP-NENs, there's such a diversity. Some people think that you should remove the GI NENs from the pancreatic NENs either way, because they can be quite different. However, perhaps more historically, NENs can be classified by their embryonic origin. And so GEP-NENs do come from the same embryonic origin. And so that's why they've often been looked at together. And at least in my project, I will be looking at them together. But yeah, broadly, the lab will be looking at NENs across the body. Yeah.
David, is there any correlation between 375 and some of these proteins that are produced by NENs? And you put your cell lines now, do those make selectivicin and the other protein?
So both BON1 and QGP1, from my understanding, are non-functional neuroendocrine neoplasm cell lines. So they're not producing the hormones. But that's interesting. In normal cell lines, or not NEN cell lines, miR-375 high expression is linked to decreased insulin expression. So that's why it's also actually being studied for diabetes. So there is a correlation between miR-375 and the hormone production. There's a potential target of miR-375 potentially targets INSM1, which was one of the transcription factors seen. So maybe there's a feedback loop there. I don't think we have currently successfully shown the targeting, but that's also one of the challenges, the heterogeneity of the literature. But it's supposed to be able to target that as well.
So I have one naive question and one more detailed. So the naive one is your literature review. You get it down to, I forget, 15 or 1300.
Yes.
And you say you're gonna extract information.
Mhm, mhm.
Does that mean you have to read all those papers and
Mhm,
extract the information, or are you using any kind of,
Yeah.
yeah.
I wish. Yeah. Well, before, so right now it's my naive list of what papers I think are relevant and will give us an insight into 375 specificity. Then I will send it to Neil, and Neil will do another look.
We're gonna cherry pick a little, so
Yeah.
once you get down to whatever, I mean, we're gonna try and group it. And then see which ones we're gonna go after, because reviews are like 50 to 100 papers, so
Mhm.
that's where we have to get to.
So your PhD thesis will have a bibliography of at least 2000 papers and something like that?
Yeah, maybe I'll give up and it'll be like two pages of my main thing and then the 2000.
So the other question though, and I think, Megan was talking about, sort of gets to it, is you have this fascinating, relationship between YAP and 375.
Mhm.
And other tumors where YAP is supposedly driving,
Mhm, mhm.
tumorigenesis, clearly something different is going on.
Yeah.
Have you, or anybody started to look at what the target genes are for presumably a YAP10, like, transcriptional regulator, in those two scenarios, in NENs versus other types of tumors where YAP is involved?
Yeah, specifically for YAP and TED, Gina, who was the MD PhD student before me, she was looking into where those potentially bind within NENs and
Within-
non-NENs
and
and
non-NENs.
and what they regulate. However, the literature is kind of divided. So, but they have seen that YAP can perhaps encourage apoptosis under different scenarios.
In
So
NENs.
they weren't focusing on NENs, or
Oh, okay.
it could be that they weren't specifically focusing on it, but the tumor they were studying had NEN differentiation because they were looking at, one was like prostate cancer, the other was breast cancer, and they can have NEN differentiation within those tumors, or because those tumors express hormones as well, maybe we're seeing a similar effect. But it was regarding microRNA 375, or not the 375 story, but the YAP inducing apoptosis within that tumor through either pyroptosis based on like whether the proteins are expressed, or due to under hypoxic conditions, YAP overexpression was actually leading to apoptosis rather than usually it can actually protect against
Okay,
apoptosis.
That's fascinating, 'cause
Yeah.
that's kind of what you might think is going on there. Other questions? Got a question? Go ahead.
It's more like in general curiosity. So why was it specifically like transcriptomic data, like mRNA chosen?
Yeah, so I think we specifically wanted to look at mRNA data for two reasons. One is, well, microRNAs target mRNAs. So we want to see what kind of the effects are happening at that level. For instance, if we see an mRNA that's high expressed, when microRNA 375 is low, then that could be a target. And if it's vice versa, when miR-375 is high, that mRNA is low, maybe that's a potential target. And that is how YAP like target was identified in the lung NEN paper. Another reason is perhaps it's giving us kind of a first view of what transcriptomic changes are happening compared to after genomic level, the mRNA level. But I do think that's a good idea to next look at the proteomic level and see if that's even a better maybe representation. Maybe those mRNAs are being targeted by those microRNAs, and now we see that change on the proteomic level or other post-transcriptional changes that might be manifested at the proteomic level. So yeah, I think that's a good idea.
All right, I don't see any more questions in the room or on the Zoom, so join me in thanking Tashifa for a wonderful talk. All right,